is the fourth experiment. The aim of the experiment is microscopic study of coronary artery with recanalized thrombosis. And for this experiment, we need to procure some requirements with a microscope, prominent slides, light microscope. It's not light microscope; it's a compound microscope. Okay, uh, and this light source we need to know. We need to arrange pencil, notebook. That we need to draw the diagram, and here we are going to know the overview of the coronary artery, which we can see the normal coronary artery, where the normal blood flow is there because the lumen is quite wide, so there is no turbulence in the blood flow. And second, here the coronary artery which undergoes atherosclerosis process, and because of that you can see the plaque formation where the cholesterol and the fat deposits takes place. Yes, Nilkamal. बार बार फिर हुआ था लेकिन अभी ज्वाइन हो गया है, you can join now. Okay, so okay. So atherosclerosis process which leads to plaque formation because of the deposition of the fat and cholesterol in the arteries. So basically, because of the development of plaque, the narrow lumen become narrow, and we can see the blood flow is quite slower or lower at the rate in compared to the normal one, right? Now the third image we can see that there is a atherosclerosis, the plaque formation, and in addition to that there is a thrombosis, thrombus formation where the clot with the plaque has done the obstruction of the lumen, and now there is no blood flow. So this is what we have seen the images, and this is what we discussed in the last experiment as well. This is the microscopic view we are going to know, and this is the three-dimensional structure. The normal coronary artery, the lumen is quite normal, lumen is wide, but in the atherosclerotic, this artery, the lumen become narrowed. You can see the narrow lumen. There may be the blockage because of the plaque. Okay, so this is the partially clogged artery, right? Where plaque we can see in the image like this, right? And then microscopic view of this coronary artery which has undergone, partially, plaque formation, where you can see the half of the coronary artery. Okay, it's having the plaque, and now the microscopic view of coronary thrombosis, we can see that this is the thrombus which has done the obstruction of the entire coronary artery lumen, right? So here there is a plaque formation in the periphery, and here you can see the thrombus formation. Okay, so thrombosis may occur followed by the plaque formation. Okay, and now we can see the recanalized coronary thrombosis, which is a rare condition. So mechanism of development of this recanalized coronary thrombosis is what we need to know. This occurs following thrombotic occlusion. So thrombotic occlusion, when there is a thrombotic occlusion followed by thrombotic occlusion, okay, this may can leads to recanalization process. So first of all, first step is atherosclerosis. Then second, there is a atherosclerosis with thrombosis. Now atherosclerosis with thrombosis may can further causes different changes. Now this is the first what we see that recanalized coronary thrombosis occurs following thrombotic occlusion. Now the thrombus which has we can see in the lumen, which attaches to the vessel wall. Okay, the blood vessel wall, the thrombus usually attaches, and the endothelial cells grow over clot to prevent further coagulation activation. Okay, so the clot which has formed in the blood vessel, Usually clot usually attached with the blood vessel wall, and the endothelial cells of the blood vessel wall usually grow over the clot, and it will prevent further coagulation and activation. Okay, it will prevent the activation of the coagulation mechanism. Now the blood vessels become leaky because endothelial cells has grown over the clot, and the blood vessel now somehow become leaky. So because there is a complete obstruction of the blood flow. Okay, I think Nilkamal is calling so many times. Let me check what is the problem with the Nilkamal. Sorry. Yes, Nilkamal. आप कैसे ज्वाइन कर रहे हो? बाकी लोग तो ज्वाइन किया, तो अब रहना तो नहीं ज्वाइन हो रहा है, तो मुझे क्लास में डिस्टर्ब मत करो, है ना? तो बाकी लोग ज्वाइन किया हुआ है, आपने ज्वाइन नहीं किया हुआ है आप। तो मैं मेरे से क्या चाहते हो? आप अपना मेल चेक करो, मैंने आपको लिंक भेज दिया हूँ गूगल क्लास। हाँ, तो सब लोग उसी से ज्वाइन किया है, सब लोग उसी से ज्वाइन किया हुआ है। नहीं, मैं करूँ क्या, बताओ तो सही। दूसरा लिंक जनरेट कैसे होगा? मैं एक बार देखूँ मैं कौन सा लिंक होता है। जिज्ञासु, कैन यू सेंड द लिंक टू द नीलकमल?
सर मैंने ऑलरेडी सेव कर रखा है ग्रुप में।
आप एक बार नीलकमल को गाइड करो ज्वाइन करने के लिए।
ओके सर।
जिज्ञासु से हेल्प ले लो, वो हेल्प करेगा, यहाँ से शेयर करेगा वो लिंक, ठीक है? So what we are learning that how this recanalization takes place when there is a coronary thrombosis process. So we need to know that the first state that this occurs following thrombotic occlusion. So the artery should have thrombus formation because of the plaque, and then after that the thrombus now attaches to the vessel wall, the blood vessel wall, and the endothelial cells, which are the cells which line the blood vessel wall, now they start to grow over the clot, which prevent further coagulation activation. Okay, and the blood vessels become leaky because of the growth of the endothelial cells over the clot, and the fibroblast grows to form scar. So here, this is the fibroblast growth also takes place, which indicates the healing process, that the whatever the abnormalities has occurred, so that is going to be healed now. So the fibroblast also grows and produces some scar-like structure, and now the recanalization to make new vessels. Now recanalization takes place so that the new vessels can be can be made because of the what the endothelial cells, and it has grown over the clot, and also the fibroblast also has grown. So both the endothelial cell and fibroblast, when they start to grow on the clot, so now the recanalization process takes place and produces a vessel like structure, and this is characterized by multiple channel formation.
एक्सक्यूज मी सर।
Yes.
सर उनका
Yes,
कृतिका को भी इशू आ रहा है और नील कमल को भी, उन दोनों का ज्वाइन नहीं हो रहा है, लिंक से भी फेल टू ज्वाइन बता रहा है।
okay. So I have no any solution. I am not IT person. मैंने आपको लिंक भेजा है, आप कैसे ज्वाइन करना है, but I don't know. Still I don't know.
हाँ
So
सर,
ask
तो
them that
यही
he will be provided. Tell him he will be provided regular lecture.
ओके सर, ओके।
Okay. So So what we need to know that the when the endothelial cells and the fibroblast usually grows on the clot, so the both of these cells participate in the healing process, and during the healing process, recanalization process takes place that produces a new vessels inside the through the that clot, and where we can see that at the last we can see that there is a multiple channel can be formed, which will establish to flow the blood across the occluded coronary segment. Okay, so the channel, multiple channel, vessels will be established at the last, and the purpose is to establish a flow of blood across the occluded coronary segment. So whatever the coronary segment which has been occluded, so through that coronary segment, the blood will be allowed to flow, and for this consequences, the multiple channel is produced because of the healing mechanism. So this is what we can see that here we have this is the lumen. The lumen is occupied by an organized thrombus. So we can see the thrombus. This is the thrombus. This area is the entire thrombus, and here also this much area is a thrombus, and outer this much is these are the plaque, and followed after plaque there is a thrombus formation. And in these two images, first image A and image B, we can see image A there are two lumen has formed, two channel has formed, whereas in image B we can see multiple channel has developed, which will allow the blood flow through each and every channel. So this lumen is occupied by an organized thrombus. This lumen is occupied by an organized thrombus, we can see, and the surface of the septa of recanalized spaces become smooth and endothelial cells grow over it. Here, this is the surface. This is the spaces. This is the space. We can see this is the space which will be like a lumen through which the blood will flow. So through this space, blood will flow, and over the surface of septa. This is the septa which divides the two space. This space, okay. These spaces are divided by the septa. Septa is like a division, okay. And this is what there is a growth of endothelial cells over this septa, and also the fibroblasts also go over this, and it makes some blood vessel like structure. So we can see the multi-channel blood vessels are developed through this thrombus. And here this is the red streak. We can see red asterisk. The asterisk shows that this is the recanalized spaces. Okay, so many spaces we can see small. So through these spaces the blood usually flows, and which tries to maintain the normal situations after the thrombosis. And this is what we have learned the recanalized coronary thrombosis. Okay, and this is what observation we can write here as the image shows narrowing of the lumen due to plaque. So first of all, we can see there is a plaque, the plaque formation surrounding the inner lining of the blood vessels, and the thrombus formation in the lumen. This is the thrombus. Okay, this much area is a thrombus. Okay, and then after inside the thrombus, the recanalization of thrombus takes place, where we can see the two canals which has developed through the thrombus, right? And interpretation we can write here is a thrombosis of it's a thrombosis recanalization in coronary artery. Okay, so this thrombosis recanalization in coronary artery is the our interpretation that this coronary artery has a thrombosis where recanalization has taken place. So this is what we have learned today. Now we can discuss about some questions related to the topic today. Now we are open for discussion now.